endocrine hormones. We have endocrine glands that are, or endocrine structures that are actually in the CNS. And then, of course, across the body, we have all of these that we'll talk about in detail later. But the whole idea of the endocrine system is to keep you in balance. And everything is about that. All of anatomy, all of physiology is about getting that homeostatic balance. We have, two big buckets to put the regulation of our daily functions or our body functions in two big buckets. One is how we respond to changes in our environment, if we have to run or if, it gets hot or other, or we eat. The insulin will help us deal with that change in our environment. Right now we have food in our stomach. That's a change in our environment. And so we eat, and hormones help us balance, help us deal with that. And then we also have, which we'll talk about in the second set, or the lecture after spring break, is the growth stages of life, like the androgens, how they are expressed and what they do to help us grow at the different stages of life. And so we'll discuss that as well. We've already sort of talked about a major hormone that we have that's released from the adrenal cortex, our epinephrine, right? Our catecholamines are synthesized in the sympathetic neurons, the preganglionic sympathetic neurons, and then the epinephrine is, the epinephrine is produced in the chromaffin cells of the adrenal gland of the cortex, and can be released in response to neural input. Sorry, I sort of skipped a step. But this is all basically what we've talked about before. These hormones are going to bind cell surface receptors and do something. Whoops, I shouldn't have said that. They don't all bind cell surface receptors, do they? We have a whole class, the steroid receptors. They go right past the membrane to the nucleus or cytosolic targets. And so we have to talk about these different receptor types and how they are transducing the signals of the endocrine system. So when we talk about epinephrine, we know what epinephrine binds to. What kind of receptors? What kind of receptors does epinephrine bind to? Adrenergic, thank you, yes, or beta receptors, that's fine. Adrenergic is sort of the global thing. So when we talk about these hormones, they're going to bind to a receptor and have something happen. And so we understand some of the processes that we've talked about in the neuro section and other places that sort of epinephrine will bind, a G protein will go and do something, maybe a channel will open, something will change. And so we often see things like where we have a spike in the concentration. In a flight or fight response or something, you'll get epinephrine up and down again. It's not going to stay up. But we have other regulation of our hormones. For example, growth hormone that will go up and stay up for stages, maybe go back down, go back up as you are growing. And then we have cyclically elevated, say for the menstrual cycle or circadian rhythms or things like that, where there's cyclical events. You'll have a spike in the concentration, then it'll go back down again. And so we have these three basic buckets to put all of these responses that we might have. Either it's a sort of quick spike, it's an elevation, or it's a cyclical event. So that's how we can sort of think of hormones in the body. And so if we're talking about the kind of the kind of cellular communication, we've talked about all of these kinds of things in other places. You've heard about these in like immunology. We talk a lot about paracrine and autocrine functions, where you can talk to the cell next to you, or you can act on yourself, so autocrine. But we are going to be talking most about the endocrine communication. So these are secretory cells. They're going to have places that are very intimate with the capillary beds. so that they can exchange or secrete hormones from the cell, and it can be absorbed into the bloodstream and carried to the organ that needs the kickstart. So why do we release a hormone? Because we need something else to work. And so we have lots of targets that we'll talk about that these hormones have. So we're going to have so many, similarities between the way the nervous system works and the way that a lot of the endocrine system works. We're going to have a stimulus that's going to excite a cell or activate a cell to do something. So we have lots of secretory cells that will secrete a hormone in response to a neural input. So that sounds just like direct stimulation of the adrenal gland, for example. So we have a lot of things going like that, but also the humoral just into the blood supply. So these messengers that we use in the endocrine system are called hormones, but we have our neurotransmitters like epinephrine. So what do we call epinephrine? Is it a hormone? Is it a neurotransmitter? And I say yes. Yes, it is. It's both. It depends on how it's used and where it's used. But basically, it is a neurotransmitter because it can excite, at the synapses. But it is also put into the blood supply, which all the other neurotransmitters don't get to do. We don't have high levels of these other neurotransmitters usually running around the blood supply. So to regulate our body, we do just like we do in the nervous system. We have positive feedback and negative feedback mechanisms that we'll discuss in different cases. We have to basically make sure that we, coordinate all the things, the cells, the tissues, the organs, to be able to come back to baseline. So we'll talk about specific examples of these as we go through. And so you can sort of do a comparison with this table from chapter 5 that sort of gives you a good summary. And this is a really important thing. The last one, the duration of the effect. The nervous system is like, bam, neurotransmitter, do something. Bam, neurotransmitter, do something. Where here, we're like putting it into this world and letting it go and seeing what it will do. And so these are having much longer lasting effects from the initial release of the hormone, because the hormone can stay around. The neurotransmitters don't last long, right? They're either chopped up by acetylcholinesterase or the type phase 1 and phase 2 enzymes of the catecholamine metabolism. Those are right there everywhere neurotransmitters are, and they don't get to go very far. We have uptake transporters so that if it sort of leaves the synaptic area, it can get sucked right back in. So we don't have that in the nervous system. But in the endocrine system, they are in the blood supply, so they can last long. They can go bind a receptor, run around again, bind to another receptor as long as it hasn't been degraded, as the hormone hasn't been degraded. Right, so this is just a little summary of the different kinds of cells, or different kinds of chemical messengers. So when we talk about amines, peptides, proteins, I don't know how to say this, prostaglandins. For some reason it's just not, but I don't say that. I say eicosanoids. Eicosanoids, that's what it is. It wasn't coming out. Eicosanoids is how I would say that, but yeah, we say it all the different ways. And so histamine, this is one of the example of just a paracrine kind of situation. Of course, the subclass is autocrine, so going across to the cell next to it, or autocrine. We have our neurotransmitters, which we don't have to worry about going through. We know exactly what this is all about, but again, I want to point out that this short and long is not duration, it's distance. and I meant to clarify that in the slide, but I obviously didn't. So the distance that a neurotransmitter can go can be quite long, right? Because it's synthesized in a neuron, and if you have a long projection neuron, it could go away from the cell body in the cortex, say, down to somewhere in the spinal cord. And so there can be great distances between where a neuron, where the neuron gets stimulated to where the hormone gets released. And so that's but this is, I said acting. That's also true, I guess, because, if you start a process, say you stimulate a neuron and you activate a G protein coupled receptor that goes and does transcriptional events, you change gene transcription, say you upregulate the level of protein of one receptor, now you have a lot more receptor on the surface. So these kinds of things can be long-term actions of bless you, the neurotransmitters. But hormones, again, are going to be released by a secretory cell, go into the blood supply, and then have to leave the blood to get to its target tissue. So that's another step that hormones need to take in order to get out of the blood and, say, hit the heart or start labor or something like that. So we have two major classes of the chemicals that we're talking about, and these are the list. We've got amino acids we consider hormones, or not hormones, but amino acids, amines, peptides and proteins, steroids, and eicosanoids. There you go. Came out easily somehow. Anyway, we can talk about the lipophobic and lipophilic ligands that we have, and so you can see their properties here. So if they're lipophobic, they're not going to like the lipids, and so they're going to be binding cell surface receptors. Almost all the time, they're going to be a G protein coupled receptor. Bet on it. So remember your couplings. Know those down, backwards, and forwards. By now, we should have them down. So the receptors are on the membrane, and you're basically going to activate an enzyme, right? What do you activate if you hit a receptor that is coupled to GQ? What are you activating? Phospholipase C, right? So or if you activate GS, you're activating adenylyl cyclase, or if you activate a whole bunch of other things. So many times, if you're activating these types of receptors, you're changing something inside the cell, so enzyme activation, or you're opening a channel, so the permeability may change. That might change, lots of things, so you can imagine. The lipophilic ligand, basically, these are going to be our intracellular receptors. Many of these would be our steroid receptors, so our androgens, our corticosteroids that we'll talk about later. They are almost exclusively, their mechanism of action is to excite or stimulate gene transcription. Yeah. Lipophobic and what? Yeah, I don't, because I'm talking about whether they like the lipids, right? So if I want to talk about how they like the water, I would use that. But so there's a proper place for the term, I think. But here we're just talking about the plasma membrane, so I'm sticking with the lipophilic, lipophobic. And yeah, so most of the time you're going to hit receptors that are going to activate genes. Genes that will, say, transcribe another hormone, or transcribe some other factor that we come across. So we have, the chemical messengers that we're talking about. These are neurotransmitters, and so we're not really going to be talking about these as part of the endocrine system, but knowing where things come from is really important. And so we're going to talk a lot about the regulation of glucose as part of the endocrine system, right? Our blood glucose is a big part of regulation of endocrine system. And so knowing that our excitatory neurotransmitters are actually synthesized from glucose, right? That's an important thing to know. And of course we have glutamate and aspartate, the two excitatory amino acid transmitters being made from glucose. And GABA is made from glutamate. And so if you don't get glucose, you're not going to get your neurotransmitters made. You're not going to be able to make all these neurotransmitters. And remember what percentage of synapses are excitatory or inhibitory amino acid synapses? 80 to 90, what was it? 80 to 70 to 80%? 80 to 90%. I can't remember. But it was a big number. It was up there near 80% of all the synapses you have are glutamatergic. You know, so we need lots of glucose to make all that glutamate. So we have to talk about the regulation of glucose, and of course brain glucose is very, very important. So we have amino acid derivatives. We're very, very familiar with our catecholamines, our norepinephrine, dopamine, epinephrine, but also the thyroid hormones are derived from tyrosine. So lots of things come from tyrosine, like the catecholamines. And we have a tryptophan derivative, so this is not a tyrosine, but it is the melatonin secreted by one of our primary endocrine glands, the pineal gland, which is located in the brain. So, you know, our amino acids and their derivatives, these amine derivatives, are incredibly important for even our, you know, not just, for our endocrine function. We have from the thyroid gland, from the adrenal gland, and from our pineal gland. 3 different sources of endocrine hormones coming from these glands, all coming from amino acids. Enough said. I don't have to say anything more about that. You guys know this backwards and forwards. And if you don't, get it down, because it's constantly coming back to you. We have lots of things that can inhibit tyrosine hydroxylase. And so, you know, in disorders that don't have proper tyrosine hydroxylase, so this would inhibit the production of all of this, right? And including, well, no, it would inhibit this pathway. We have lots of peptide hormones. We're going to spend our time, most of the time, talking about this. Most of the hormones that we'll discuss, like the ones that are on this slide, are peptides, small peptides, maybe, you know, not a big protein, not more than 20 amino acids or so. No, 50, not more than 50 amino acids or so. And so things like oxytocin, we have bigger ones that are more like proteins, like insulin, growth hormone, and prolactin, that are much bigger. But most of the things we're going to be talking about have this sort of small peptide function. And we learned a lot about peptide neurotransmitters in the neuro chapters, right? And what kind of receptors do peptide neurotransmitters bind? It should be like G protein coupled receptors. G protein coupled receptors. So all of these things, many of them, not the steroids, but many of them are going to be binding them. Just don't ever forget that. And I won't let you, I guess. I'll remind you all the time. So most hormones are these kinds of peptides, or they can be bigger, small proteins, not really big proteins. We have glycoproteins that are basically polypeptides with carbohydrate side chains. And so we have things like insulin would be considered a glycoprotein. So it is a small protein, but it's got these carbohydrate side chains as insulin. And then we have other things like TSH, LH, FSH. These are all these small peptides or glycoproteins, or small proteins. And so most of them go through this process, So do our peptide hormones. You remember the sort of enkephalin synthesis. It's, it basically chops it down, chops one big gene product down into little pieces until it gets the different products for the endogenous endorphins. And here is the same way. You get a pre-pro hormone, a pro hormone, and then finally your hormone. And of course, it would go through a process that we're very familiar with. We understand how mRNA is translated into a polypeptide, right? That happens at a ribosome. The ribosome translates the RNA into protein. The protein is transported through the endoplasmic reticulum. It can get folded into the right shape because proteins have a three-dimensional shape, and so when a protein goes into the ER, it's in its primary structure, and when it comes out of the ER, it's got secondary structure. And so then it gets packaged into the Golgi, or transported into the Golgi, and the Golgi will direct its release at the surface of the cell. And so as it passes through here, it goes through these stages. So it's not a full hormone in the ER or even the Golgi. It doesn't happen until it's in that final vesicle. So because we don't want a lot of extra, fully prepared hormone running around. We want it to be, made when we need it, and then sometimes some are stored, but usually we make it when we need it. Get a signal that says, okay, start making this particular mRNA. The mRNA gets transcribed by polymerases in the nucleus. Then we get the transcript, the mRNA, and so on. So we know all of that. So that's how we get those peptides out and they're released. And we actually know how we might be able to stop that by stopping the synaptic vesicle fusion. Just a thought. Anyway, our steroid hormones, they all come from lipids. And I really want this table to move because I can't stand over here. So hold on a second. It's in my way. I should just take off all the little stoppers. I'm a Taurus and I'm stubborn, so I'll just keep pushing it. Okay, so yay. I can stand over here now. So beat that. So we have lipid hormone or steroid hormones that are derived from lipids, and so we have most of what we'll talk about today are cholesterol derived. We have others that are derived from there we go again. Eicosanoids are also lipid derived. So the steroid hormones coming from cholesterol. So cholesterol is broken down, and you can see at different stages, we have lots of inactive or not active intermediates, but so, sorry, distracted. And so we can see from cholesterol, look at everything we can get: progesterone, cortisol, estradiol, testosterone. So all of our sort of, famous or, most familiar hormones are synthesized from a lipid in our membranes, cholesterol. So I find that absolutely fantastic. These steroid hormones can be released by our reproductive organs, like testes and ovaries, or the adrenal gland from our cortex. The medulla is where the chromaffin cells are, and that would be the epinephrine source. And then we have a completely different layer, what I'm going to go through later, that is the cortical layer, which produces the corticosteroids. And then the kidneys, of course, will be able to produce calcitriol and help with calcium regulation in our from the proximal tubules. So we have these steroid hormones that can be released from all of those. Most of the time we find them bound to plasma proteins, so they can remain quite a long time in the circulation compared to some of our peptide hormone friends like LH, FSH, and TSH. Usually those are pretty quick time courses, relatively speaking, not on neuro scale, but endo scale. Here you can find some of these things running around with albumins and other globulin proteins, globular proteins. So then we have the eicosanoids. These are also derived from lipids, often from arachidonic acid. This is a membrane phospholipid, close parentheses. Again, as I didn't mention here, but as I have it on both slides, these are synthesized on demand. So we also have some neurotransmitters we found to be synthesized on demand, right? So nitric oxide, the cannabinoids, these are also synthesized from lipids. And so very interesting, world we live in, that all these things come from just one or two building blocks, amazingly. So we have our arachidonic acid, and through the action of another phospholipase called phospholipase A2, can break down membrane phospholipids into arachidonic acid, and then through two different pathways, the lipoxygenase pathway, we get leukotrienes, or through the cyclooxygenase pathway, we get prostaglandins, prostacyclins, thromboxanes. So these are eicosanoids. These would be considered the eicosanoid types that come out. Right. So leukotrienes can play secondary roles, but prostaglandins will regulate local inflammatory responses in wherever they're getting produced. Yes?
Are the peptide hormones also synthesized on demand, or is it just the lipid-derived hormones?
So it depends. Some of the peptide hormones are stored. Many of them are stored in the, I don't actually know exactly if I can say where. I mean, some of the tissues, some of the primary endocrine tissues actually store some of the peptide hormone, but I honestly can't be 100% on which ones are which, but I will check that out. Yeah, I don't actually know. Right, so if we're going through the blood, we're thinking of our hormones in the blood in two ways, either dissolved in the blood itself, carried around easily just with all the rest of the blood cells, or bound to these carriers where most of the most of the hormone is bound to the plasma protein and would vary, that concentration, the free concentration would vary with the levels of plasma proteins you have. And so whenever we're talking about a compound or a drug or an effector molecule, we can talk about its half-life. And so we can talk about these chemical messengers, these things these hormones that we're talking about to have the different half-lives. And so if we're dissolved in the plasma, like the lipophobics, then basically we tend to have the shorter half-life. So insulin, for example, does not stick around very long. It comes on, it gets increased in concentration, and then that drops off. And then we have things like cortisol, where the half-life is 90 minutes compared to the less than 10 minutes for insulin. So it can make a huge difference. So 10 minutes to 90 minutes is a big difference in time, or half-life of the drug. And so what is the definition of a half-life? Yes. Okay, yeah, so if you're thinking about elimination kinetics, we would be talking about how the time it takes for the concentration to drop by half. So that would be half-life if you're speaking of elimination kinetics, but you also have accumulation kinetics to think about too. So, you know, the time it takes to get to 50% of its peak concentration. Both ways of thinking about it. So that's this way. The peak concentration thing is this thing. Thinking about half-life in this way. So, you know, how does it? How do we get to? Our peak effects. So we can look at different factors that will influence the strength of the response that the hormone may get. And so, you know, there's a certain amount of hormone released from a certain cell, and it's going to travel around, and it's going to have a certain concentration. And so we can predict its effects by using these kinds of graphs. And so if we know what percentage of the receptors are bound, we would say that when 100% of the receptors are bound, we have this would be a concentration that would allow us to get to maximum efficacy. So we have every receptor that's there that we can use activated. That's as good as you can get. And we have to define that as 100%, right? No matter what, no matter how many receptors are in the tissue. If you only had 5 receptors, if you hit all 5 of them, you've reached maximum efficacy for that particular receptor system. And so we can look at receptor bound versus concentration. We can look at one concentration and then doubling that concentration, or doubling the number of receptors that we have bound by the drug. This drug can only bind about 50% of the receptors that we have, and this drug can bind twice as many receptors, and so we would expect a bigger effect out of this. I don't mean to say drug, I mean to say hormone, because we're talking about hormones, but same idea. And then if you actually look again at the percentage of receptors bound over concentration, and we have 2 different hormones, the hormone that can reach the 100% sooner would be the stronger or more efficacious compound. And so when the affinity, which is defined as the concentration at 50% of the effect, and so our effect in this case is how many receptors are bound. So here's our effect, and we want to know the affinity. We just ask, here's our curve, the orange curve. We're doing orange curve. Where, it reaches 100% finally over here, however long it took to get there. And so we ask, what concentration is at the 50% mark? And so we draw a line over to 50%, and we come down, and we see the concentration is, say, 100. We look at the red line, and we see its peak concentration is the same because they can hit the same number of receptors. So the receptors aren't limiting this situation. What's limiting is the affinity, or the strength of the binding interaction between the hormone and its receptor. And so here, 50% of the peak effect, the 100, 50% of that, we go down, we have a much lower concentration. So if this is 100, that was 10, just making numbers up. But we only need 10 of this compared to 100 of this to basically get the same effect. So we worry about the efficacies of these compounds at the different receptors, and all the receptors will have different efficacies for the compound. So for example, a luteinizing hormone, I believe only has one type of receptor, but if it had more than one receptor, there might be different actions at that receptor depending on its affinity for those receptors. Epinephrine has various variable affinity for the 3 different adrenergic receptors, for example. So this is all about our strength of binding, and then strength of binding translates into the magnitude of our effect, of the signaling effect that we get. So, or the cellular response, what's coming out of this. So we have different scenarios that we can think about. Usually when we're talking about binding of any kind of, biological or bioactive compound, they usually bind quickly and release quickly and go away. And then they might bind again for something else. But these are typically reversible events, and so you might have bind, the luteinizing hormone, as long as it's sticking around in that area, can continue to bind, continue to activate until it's washed away or degraded or whatever happens to it. So we might have one messenger that can bind to lots of different types of receptors, and so you have one stimulus that releases the hormone into the body, but then it can act on that type of receptor and that type of receptor and this type of receptor, and have a very large effect in the body, the number of receptors can be very different. And so one tissue that has only a few receptors, one tissue that is loaded with them, will have different strength of the response to that particular hormone, for example. One target may have many types of receptors, and so, say you've got a hormone, a gland or something, and not just one class of hormone would activate that gland. So we have lots of different scenarios that we can look at. Our upregulation and downregulation, this is going to be, again, talking about gene transcriptional events. Do we increase the transcription of, say, that particular receptor so that we can detect more luteinizing hormone? I don't know why I'm picking on that one, but I just am. And so, if we don't have enough of that, it says, the body says, hey, I need more of that. And so we start to make more. We send transcription factors into the cell, make some gene transcriptional events so that we get more gene product of the receptor expressed on the surface. And of course, you can go the other way, you get downregulation. So now if you're taking receptors off the surface, you're not going to be as sensitive to the drugs. Like, you put on the drug, you're like, what's going on? Why aren't you responding? And then 10 times more. What's going on? Because downregulated. Or there's a tolerance. So the way that receptor responds to the drug has changed. So I'm not sure, I haven't done that with you, but maybe I have. Should we talk about the phosphorylation of the beta-1? I don't think we did. But I'm going to tell you a story anyway, because this is part of receptor pharmacology, is that in repeated stimulation of receptors, there is a situation in the betas in which repeated stimulation of beta-1 receptor with norepinephrine will cause not just the normal pathways that you would expect, the GS pathways, but then if that is continuously activated, a separate pathway activates protein kinase C that comes over, phosphorylates beta-1. Now beta-1 can only couple to GI. You flipped beta-1, and it's no longer coupled to GS. It's now coupled to GI because you continuously, activated the norepinephrine pathway so much that they're saying, enough of this. You've activated this whole weird pathway that activates protein kinase C, which is a kinase which will phosphorylate. And what that kinase does is comes around, phosphorylates that same beta receptor, and a phosphorylated beta receptor couples to GI, not to GS. I think this is a very, very cool story. Not cool for the people who have their beta receptors switching sides to the GI, but it's still this, too much signal can cause a tolerance, and that you're not changing the expression of the beta, it's still there, It's just phosphorylated and acting completely different than it was. So a very cool mechanism. An example that we can see for the insulin receptor, for example, the skeletal muscle wants to be able to regulate glucose, right? And so you put more insulin receptor on the surface, you get more absorption of glucose in the tissues. So, right, so we've talked about G protein coupled receptors, we've talked about channels, but we didn't talk about a lot of the other types in any specific way. I might have talked sort of on the side about them, but in our membrane, we can have basically three types: the channel linked, the enzyme linked, and G protein. And so we defined these a little bit earlier, like the fast acting and then the slow and the very slow, when we were talking about initially introducing receptors in the neuro section. And so these are going to be your types of receptors that you'll see in when they bind the plasma membrane. And usually we're going to do something. We're going to open a channel, activate adenylyl cyclase, or do whatever it is we're going to do to affect that. So these are channel-linked responses, or not channel, receptor, transmembrane channel, it's not channels, transmembrane receptor protein, that's on the surface there. And of course, they are going to be regulated all by the second messenger systems like cyclic AMP or calcium. Then of course we have our lipophilic ligands that are going to go right past the plasma membrane and find the intracellular receptors. And so we have a whole slew of intracellular receptors that live in the cytosol. And so they don't have to go far, they just have to go over the membrane, and there you are, you can find your receptor. But some of those receptors are in the nucleus, so they have to go past the nuclear membrane as well and get all the way into the nucleus to find and bind their receptors. So steroid hormones, thyroid hormones, that's the type of receptor you would see there. So we had our fast-acting channels. We're very familiar with this. I'm not going to spend any time talking about this. And channel-linked receptors, where you can have this is basically the same thing. Channel. So but we have calcium. Fast ligand-gated calcium channels. We haven't really talked about ligand-gated calcium channels. We talked about the voltage-gated calcium channels in the synaptic boutons, but not ligand-gated calcium channels. So we see this in certain tissues, and especially where calcium is important, like where we have to do contractions, like muscle. Smooth muscle, skeletal muscle. One of the things that is really important is this pathway, this other second messenger pathway. So calcium is sort of like the quintessential second messenger. But calcium goes right on to talk to calmodulin to get most of its work done. So by itself, calcium doesn't do as much as it can, but for the calcium calmodulin. So there is this entire group of enzymes known as the calcium calmodulin activated enzymes. So when you have calcium and calmodulin together, they can activate a whole bunch of enzymes. And so that's what this sort of thing is that when this is together, when calcium and calmodulin are allowed to be together, they can activate tons of enzymes, which usually will result in tons of phosphorylation events, either intracellular, proteins or at the surface. Yes?
This is mostly happening in like skeletal muscle?
In muscle. Yeah. Yeah. A lot of this happens in smooth muscle, particularly the calcium calmodulin pathways. Okay, and then one of my favorites is the kinase linked receptors, or phosphatase linked receptors. So we talked about kinase a lot. What's a phosphatase? Who knows? Phosphatase. Tase that phosphate. You take the phosphate off. Kinases put phosphates on, phosphatases take them off. So it's sort of the yin and yang of phosphorylation here. And so phosphatase reaction would go this way. The enzyme would take the phosphate off the protein. So many times phosphorylation will activate. So if you're phosphorylating something, this could activate that receptor type, and then that receptor sitting in the membrane has an intracellular tail that's now a kinase, and it can activate all kinds of things. And in fact, it's often activating things right next to it, like lipids that can turn into steroids, for example. So just kind of pulling that loop together. We have a lot of different types. We have the tyrosine kinases. Those are the famous ones. There's a whole slew of drugs right now blocking receptor tyrosine kinases that are really have been really beneficial in clinical trials, and so we're very hopeful for our tyrosine kinases. But they have also phosphatases, and it's not just tyrosine kinase. You can also have serine threonine kinases. So like the proteins I study, the bone morphogenetic protein receptors, BMP receptors, they are serine threonine receptor kinases, not tyrosine kinases. And so they will phosphorylate things on a serine or a threonine. And tyrosine kinases put a phosphate onto a tyrosine. So that's why they're not very inventive. So tyrosine kinase will put a tyrosine put a phosphate on a tyrosine, and a phosphatase will take it off. Specifically the tyrosine or serine threonine. We now know that there's quite a few enzymes that have both activities, tyrosine and serine threonine. We thought they were really separate sort of evolutionary things, but now we see dual action kinases that we're coming to know. There's a third type of kinase, the guanylate cyclase, not kinase, but enzyme-linked receptor. So most of our enzyme-linked receptors is either going to be a kinase or a phosphatase, but we do have the guanylyl cyclases. So guanylyl cyclase sounds just like adenylyl cyclase, except it starts with a G, right? It's very similar and does the same thing. Adenylyl cyclase converts ATP into cyclic AMP, and guanylyl cyclase converts GTP into cyclic GMP. And so there are a number of neurons that operate on the cyclic GMP pathways instead of cyclic AMP, under the control of cyclic GMP rather than cyclic AMP signaling. So we do have cell membrane, transmembrane receptors that have, instead of a kinase in their intracellular domain, they have this guanylyl cyclase that can convert GTP into cyclic GMP. So this didn't reproduce very well. This happens to be the pathway that we know for the insulin receptor. And so the insulin receptors are tyrosine kinases. They are transmembrane receptors, sit in the membrane, and have a cytoplasmic tail that gets phosphorylated. The phosphorylation activates it. And it turns out that they need a dimer in order to work. And it turns out that the dimer comes together, and this one phosphorylates this guy, and this guy phosphorylates this guy. It's called transphosphorylation, and they activate each other in a dimer. And that dimer is required to interact with these adapter proteins. So these adapter proteins called GRAB and SHIC and a whole bunch of other things, which aren't important for us to know here, can link this insulin receptor to intracellular signaling pathways leading to nuclear transcriptional events down here in the nucleus, for example. So that's what this is showing. So insulin is going to cause changes in gene transcription to give you the cellular response that you want. And so don't need to worry about all these guys, but these are just the known guys. These are known as the small G protein families, RAS and RAF, and then these are MAP kinases. This is MAP kinase, and MAP kinase, something like that. Anyway, they're all a bunch of kinases continuing to phosphorylate each other, and the next one leads to the next one, and it's these loops. And so you can have amplification here, which is really important in signaling. Right? One receptor binding event can result in many things happening because you're not just activating one RAS, you can activate 10 RASes. And then that 10 RAS can activate 100 MECs, or RAFs, and then RAFs can activate, so it can build, right? I think there's an example coming up of something like that. And so, yes. So this would be enzyme linked. So it's a major part of signaling in the cells from chemical messengers. But for us, again, we're going to spend a lot of time either on the steroid receptors, the intracellular ones, or these, the G protein coupled receptors. So here are your old friends, the second messengers, and see we do have second messenger systems that rely on cyclic GMP, so having the guanylate cyclase. Sometimes it's soluble in the cytoplasm, a soluble guanylate cyclase, but then we also have the transmembrane type of action. And so they each activate their own thing, and you can see epinephrine, for example, will activate beta 1s and create cyclic AMP. And so you can see these other ones here. So know your pathways, understand what GS does. GS will activate adenylyl cyclase, convert ATP to cyclic AMP, cause the activation of protein kinase A, which goes on to do tons of different things. So there's this video, which isn't bad. It's kind of nice to watch how cyclic AMP does it. So I encourage you to watch it. And then this is supposed to be our phospholipase C. And we've looked at phospholipase C pathways, the GQ pathway, before. So this is not new. But it is showing you a couple of extra things that we hadn't really mentioned before, or it's just on the slide, but we didn't really pay much attention to. And so for GQ, we're going to have the activation of a different enzyme. That's phospholipase C, not adenylyl cyclase. Now it's phospholipase C if you're talking about GQ. And it will take these lipids that are right here and lipid products, so this phosphoinositol 2-phosphate, are converted into IP3 and DAG. So IP3, we know, goes back to the endoplasmic reticulum and binds to the IP3 receptor, which is a calcium channel. And that calcium channel allows calcium to come out, increase intracellular calcium. The calcium calmodulin pathways can now be activated. And so we have our calcium calmodulin here. That's supposed to be calcium, the green. It can do other things, obviously. You can go sit and talk to troponin and get some sliding action happening at the filaments, but it also does quite a lot of this. So protein kinase C, or protein kinases, and then on to its effect. The one we have not spent any time really talking about are these, the intracellular receptors. So all of these are going to be like these lipophobic. They're going to pass through and activate intracellular receptors. And so you'll find these receptors either in the cytosol or the nucleus. And so in this example, we see the receptor is in the cytoplasm, but there are receptors that are in the nucleus that will bind directly. They'll go right from being here right into the nucleus, because that's where this receptor is. And so steroid hormones are the big ones that do this, and they make these complexes of the receptor and the ligand. If it's in the cytosol, the whole complex will travel into the nucleus together as a complex. or if it's already in the nucleus, it doesn't have to go anywhere. But the main goal is to regulate genes. And genes have all these little regulatory elements in them. A lot of them are before the start codon, right? So upstream of the start codon. And in there we have the promoter regions, enhancer regions, but we also have these response elements. And so we have things like hormone response elements in genes that respond to hormones. So if you're a gene that responds to a hormone, you're likely to have a hormone response element upstream of your start codon. And that means that you'll have the hormone will activate transcription of that particular gene that is supposed to be activated in response to that hormone. And it can affect that transcription. So it could repress it, doesn't have to activate it. It could repress that, and so that you don't get more expression, or you try to make the minimal expression. So rate and patterns of protein expression can be, this part of the pathway can be regulated by the binding of the HREs. And if you're a glucocorticoid, you bind a GRE, and if you're a corticosteroid, you bind a CRE, and so they have all of these different names. If you're a BMP, you bind BMP response elements. So everything has a response element. Not everything, but lots of these regulated genes do, especially genes that produce hormones. So this is the scheme for a intracellular receptor. They also form dimers, and a dimer is required for full activation of the response element. And so these we're not worrying about whether they're in the nucleus or the cytosol. They're going to be in one of those places. In the cytosol, it'll go to the nucleus. It's got to get to the nucleus because that's where the DNA is. So it's going to the DNA. Here's our pink DNA, and right here is the HRE. It's got a tandem binding site because we need two receptors for full activation. So each steroid receptor, for example, has 3 domains. There's the Pac-Man head, that's the ligand binding domain. There is a linker region called the regulatory domain, which has a cool role, but I'm going to go too far into the weeds if I do that. And then DNA binding domain. So the DNA binding domain, we know where that goes. It sits on the HRE. So these DNA binding domains are going to interact directly here. So once the steroid hormone binds the ligand binding, you can dimerize, and then that dimer will fit properly, have the proper affinity to bind to this region of this DNA. And this DNA will result in some transcriptional event, in something, I don't know what it is, but it's something. And so we see that here for steroid hormones, and so like I said, for our glucocorticoids and mineralocorticoids and all the other corticoids, but also for our thyroid hormone receptor. So the thyroid hormone also has this 3-domain structure of binding a thyroid hormone binding domain, the regulatory, and a DNA binding domain. But in order for thyroid hormone to work, it has to dimerize with the retinoic acid receptor. And I almost forgot what it was. Blank. So retinoic acid, there, it would have reminded me. Retinoic acid binds to the retinoic acid receptor and dimerizes with the thyroid hormone receptor to produce mRNA. So it binds the T4 and allows the transcription of thyroid-related genes, thyroid hormone-related genes. So an interesting thing about the thyroid hormones is that their receptors are located in 2 places, and they kind of have 2 different functions. So we get thyroid hormone actually transported across the membrane through a carrier-mediated process, because it's not going to want to go through there. And what it does is it can either bind to receptors that are on the mitochondria, and that's a direct relation to generation of ATP. So it basically kickstarts the mitochondria, says, hey, make some more ATP, we need energy. So it can bind directly to cytosolic receptors. Well, they're not really cytosolic, they're mitochondrial receptors for the thyroid hormone, and it can increase energy. And so, sorry. And then it also has its typical nuclear receptor. So thyroid hormones have their receptors in the nucleus or on the mitochondria. And then, of course, if it sits on the nucleus, it's going to be dimerizing with the retinoic acid receptor, using retinoic acid and thyroid T4 to stimulate thyroid hormone gene transcription. So then you can just see all these different, what their function is, basically. So when it's a G protein coupled receptor, it just says that because it's that whole pathway of either phospholipase C or adenylyl cyclase activating, but you also have some channels like the glutamate and the serotonins. So the interleukins, which is a major immune system peptide cytokine, those all use the enzyme linked receptors. And insulin is also an enzyme linked receptor. It's a tyrosine kinase receptor. Yeah.
For the thyroid hormones, they're not lipid based, but they're still lipophilic.
No, they're still lipophilic. They get carried across the lipophobic. They still get carried across the membrane, like through a channel or something. They don't diffuse through the membrane. So the steroid hormones do, not thyroid hormone. Okay. So I neglected to put page numbers on these slides. I don't know where we are. And so it's 3 o'clock. Why don't we take a few minutes break?